We are now going to shift gears a little bit away from the activation of T cells toward the activation of B cells. As Dr. Alley talked to you about in an earlier chapter, mature B cells are going to leave the bone marrow after they have gone through their development, their rearrangement of their immunoglobulin genes, selection. They are going to recirculate throughout the body, again, attracted to the follicular areas of the secondary lymphoid tissue. And just remember that this increases the chances that these cells are going to come in contact with antigen that they may be specific for. As Dr. Alley mentioned, for T cell activation, B cells also require more than one signal to become activated. Once the antigen enters the secondary lymphoid tissue, if the B cell is specific for that antigen, it can bind to the B cell receptor, which, much like we talked about with T cell activation, provides the first signal for activation. What we need to discuss, however, before we really sort of get into B cell activation are the two different types of antigens that B cells can encounter. And these are the thymus-independent, or the TI antigens, versus the thymus-dependent, or the TD antigens. So let's take a quick look at the difference between a T-cell independent antigen versus a T-cell dependent antigen. T-cell independent antigens can activate B cells, just like the name suggests, without the help of T cells. There are different sorts of molecules that can do this. One example, is a really good example, is lipopolysaccharide. Lipopolysaccharide has a lot of repeating subunits and just can sort of lay itself over multiple B cell receptors, which can result in activation of the B cell. Now, without the help of a T cell, the antibody isotype that a B cell can make is IgM. And that's going to be really important. That's actually going to be an important concept throughout a lot of the things that we're going to talk about, especially when we start to bring in some of the microbiology concepts as well. And I'll talk about that in just a little bit. If we compare that to a T-cell dependent antigen, we have a lot more that's happening as far as activation. First of all, you have to recall that T cells can only recognize peptide. So any antigen that's going to be a T cell dependent antigen has to be a peptide, because T cells can only recognize peptides, versus B cells, which sort of handle the bulk of really everything else. So they can handle different molecules such as polysaccharides, lipids, etc. When a B cell is activated by a T cell dependent antigen, it processes and presents that antigen just like we talked about in a previous chapter and puts it on a class 2 MHC molecule for presentation to a T helper cell. And there are other activation signals that we're going to talk about in just a little bit that not only help the B cell become activated, but also in turn activate the T cell. Remember, a lot of the times with immunology, these cells are really trying to help each other out. So it's sort of like a you scratch my back, I'll scratch yours scenario. So let's look at a couple more details about the independent antigens, and then we will walk through the T cell dependent antigens. So as I mentioned in the previous slide, T-dependent antigens have to be peptides, but T-cell independent antigens could be just about anything because they don't have to interact with the T helper cells. These T-cell or thymus-independent antigens, as we mentioned before, directly stimulate B cells to proliferate and differentiate into, and here's the important part, IgM secreting plasma cells. This is going to be sort of an important concept, not necessarily just when we're talking about T-cell independent antigens, but thinking about in different situations when we have a lack of T cells, what type of antibody is capable of being produced. And there's two situations I can sort of think about right offhand that step one would potentially ask you about, and that is in a patient with AIDS, where you're actually lacking the T helper cells required to cause class switching. What types of antibodies can you still find in those patients at near normal or sometimes above normal levels? That's going to be IgM, because remember, we don't need T cells to produce IgM. Another good example where they can sort of ask you something like that is if they were talking about a patient with DeGeorge's syndrome. And we will get to that in a future chapter on immunodeficiency diseases. But of course, in DeGeorge's syndrome, those patients are also lacking T cells. So the same sort of scenario. So those things can come up in clinical vignettes, and you would be expected to know, of course, that the only isotype you're really going to have in any kind of concentration without T cell help is going to be IgM. Now, there are certain things the B cell has to give up if it's not getting the help of T cells for activation. And the first thing is that the overall response to a T cell independent antigen is generally weaker than the response to T cell dependent antigens. And again, we typically are looking at secretion of IgM versus all of the other isotypes that we could potentially get with a little bit of T cell help. The other sort of really important concept when we're talking about T cell independent antigens is that there is no memory response. So we are lacking that memory response that we need. Now that is also going to be an important concept when we get to the chapter on vaccinations, because if we're looking at a vaccine that is made up of nothing but capsular polysaccharide, if we're using a vaccine, again, that's just capsular polysaccharide, remember, T cells require peptides. We are going to get an IgM response, and we are not going to get a memory response per se. So we're not going to have booster dosing and things like that that we would normally really look for in a scenario where we're talking about vaccinations. So those types of vaccines, and really there's one major one we use now, which is for strep pneumo in usually adults over 65. We're really just trying to protect them against multiple different serotypes of a very, potentially serious disease. So those are all really important clinical concepts that may come up on step 1. B cells that are activated by these T cell independent antigens are found typically throughout the spleen and the mucosa. Most antigens that B cells will interact with are actually thymus-dependent or T cell-dependent antigens. This response is going to give a much more robust development of antibodies. We will actually get isotype switching, but it requires a couple of different interactions that are going to be really important. One of them is, of course, the CD40-CD40 ligand interaction. We mentioned this in an earlier chapter, and Dr. Ali sort of previewed it for you that it was going to be important in a later chapter when we started talking about T cell dependent B cell activation. So let's take a look in the next slide of essentially what the important steps are in this sort of B cell activation. So before we start this, I want to walk through with you and remind you that Dr. Ali at the beginning of this chapter sort of talked about an interaction between T cells and macrophages, where you could see the interaction between the T cell and the macrophage, what sort of molecules the T cell was looking for by way of the pro-inflammatory cytokines, by way of the B7 molecule, and then what sort of things the macrophage was looking for, in particular that interferon gamma. A lot of those same features are here when we're talking about T cell dependent B cell activation, but what the B cell relies on a little bit more than some of those other cell-cell interactions between T cells and other APCs is going to be the CD40-CD40 ligand. So let me just walk through this with you really quickly. B cells, recall, are the only APCs that specifically bind to antigen through their B cell receptor. And remember, they're an antigen-presenting cell, so they will bind to the antigen, they will process it, and present it on a class II molecule for the presentation to a T helper cell. We already saw this other interaction when Dr. Ali talked to you about the macrophage T cell interaction, and that's the B7-CD28 interaction that the T cell really needs to become activated. That's its major, really co-stimulatory molecule. But what the B cell is really looking for is this CD40-CD40 ligand interaction. Now, this is going to be a really important interaction, so you want to really keep your eye on that because, of course, deficiencies in the CD40 ligand can contribute to immunodeficiency diseases. We also require that interaction for certain types of class switching. It is also required for memory and also the formation of germinal centers. So that interaction is going to be really important within that B-cell, T-cell interaction. Once those molecules sort of come together, the T-cell is going to provide cytokines like the ones that Dr. Ali talked about for the differentiation into the T-cell subsets that are going to tell the B-cell to class switch. And it really is dependent on the right combination and cocktail of cytokines that are made as to which immunoglobulin isotype the B-cell will switch to. Now, what's really sort of neat when we're talking about B-cell activation is that these B-cells that are activated are released in two waves. And this is something that you probably never really thought of maybe, but you already really sort of know this because we know that if we have an acute infection, the antibody that we're going to make predominantly is going to be IgM. So that makes perfect sense. The primary wave of activated B cells is comprised of those IgM secreting plasma cells. Remember, we're always going to make IgM first. Those plasma cells are going to leave the secondary lymphoid tissue shortly after they are developed and activated. The second wave of activated B cells, though, is going to stay within the secondary lymphoid tissue and just get tweaked a little bit. They are going to develop higher affinity antibodies that are going to go into creating those memory cells. So the memory cells that stick around are going to be much better at binding antigen. But again, this is sort of the second wave. This is now going to involve antibody isotypes that have undergone class switching. So in the next couple slides, we're going to talk about this process of affinity maturation, and then we will finish up by talking a little bit about class switching. Remember, just put yourself anatomically in the host as to where these things are happening, and recall not only just in the secondary lymphoid tissue, but in the germinal center. Remember, we said there were three really important events that happen within the germinal center: affinity maturation, class switching, and then, of course, the decision to become either a plasma cell or a memory cell. All right, well, there are lots of cool things that happen in the immune system, but affinity maturation is really one of those really cool things. It's like we have all of this incredible amount of diversity, of this incredible immune system that can somehow kill things that aren't supposed to be there without killing us if everything's going well, okay? But what happens during an immune response is during the time that these cells are actively proliferating, and it is extremely intense. Think about eukaryotic cells dividing and proliferating about as rapidly as they possibly can. The DNA polymerase is completely overworked. It's stressed out. It's normally a very type A enzyme, right? And it likes to proofread. It sort of reads the template, it puts the appropriate nucleotide in, and if it screws it up, it typically proofreads and will excise out the bad nucleotide, put in the correct nucleotide. Well, during this intense proliferation where we know we need cells fast, we do get random mutations that the DNA polymerase really can't handle. And these mutations occur in the variable region of the immunoglobulin molecule. This process is called somatic hypermutation, where we get these point mutations in, recall, the idiotype of the antibody, which is created by the variable regions of the heavy and light chain coming together. If these mutated idiotypes have an increased affinity for the antigen, they sort of have a selective advantage over everyone else. And we'll talk about how that somatic hypermutation then results in this really cool process called affinity maturation. So let's take a look at how somatic hypermutation can result in antibody molecules or B cell receptors with a higher affinity for antigen. The binding antigen serves as that first signal for proliferation, and if we're getting these point mutations within the already rearranged variable regions of the immunoglobulins, you can have really just a couple of scenarios that might happen, right? It could negatively affect the binding of that B cell receptor to antigen. But let's think about what might happen. If we lowered the affinity for antigen, that B cell is not going to bind antigen as well or as easily. And eventually, if the B cell doesn't bind antigen, remember what happens to it. It's eventually going to die off. However, if we result in a higher affinity B cell receptor through this process of somatic hypermutation, they're going to naturally bind antigens with higher affinity. And what we're actually seeing now is something called clonal selection, because those B cell receptors bind antigen better. And every time they bind antigen better, they're sort of ensuring their survival by being activated themselves and proliferating. So the new pool of cells that we're generating has a much higher affinity for the antigen based on this process. And our memory cells are coming from this pool of mature cells as far as this increased affinity. The last concept we're going to talk about in this chapter is isotype switching or class switching. Before we get into that, we sort of have, again, this antibody molecule that you've seen once before. I just want to go over, we just finished talking about somatic hypermutation and affinity maturation, and I just want to reiterate that process occurs in the idiotype. So again, the variable regions of the heavy chain and the light chain, and that's going to affect the binding of antigen. What we're now going to talk about is switching of these constant regions of the heavy chain from an IgM and potentially an IgD to the other isotypes IgG, IgA, or IgE. So we are switching the constant regions of the heavy chain to different isotypes, and that's going to change the function of that antibody molecule. Remember, the isotype dictates the function of what that antibody can do. And we're going to talk about that a little bit more in the next chapter, so I'm not going to give too much of it away, but it's an important process so that we can gain some additional functions that can help us battle these pathogens. So as we mentioned before, once the B cell receives its signal to class switch, and it's going to get that signal from the T helper cells, it is now going to work on switching the constant regions. Remember, these constant regions are downstream from the variable regions, which we rearranged during development. Okay? Now, the only thing that's going to happen to those variable regions once we come in contact with antigen is potentially they're going to get a little bit better. Okay? Now, though, we get to switch the constant regions, so we can go from expressing this IgM and IgD to one of the other constant regions. And remember, when we change that constant region, we now have potentially acquired new effector functions. Okay? Just like we mentioned before, what happens is if we are going to, in this example, we are now creating the isotype IgG, but this is specifically going to be an IgG1. And we'll talk about this when we talk about humoral immunity, but there are several different subspecies of IgG. There's IgG 1, 2, 3, and 4, and there's actually 2 subtypes of IgA. But in this example, we're switching to an IgG1. The intervening DNA with some other constant regions is looped out and removed, and now this B cell will forever be a B cell that's expressing IgG1. Now, what's important for you to know is that whether this cell is a memory cell, whether this cell is a plasma cell, the receptor on the surface now shifts from being both IgM and IgD to whatever we class switch to. So we're going to make an IgG1. That is now going to be the B cell receptor. And if that memory cell comes in contact with antigen that it's specific for, which of course we hope it does, it will differentiate into a plasma cell that also makes IgG1. So that is a one-way switch. And again, with class switching, we are changing the effector function of that antibody molecule. Isotype switching or class switching, as Dr. Ali sort of alluded to already when she was talking about the different T cell subsets, is dependent on T cells and different combinations and or cocktails of cytokines. What happens in class switching or isotype switching is very similar to the process that we already talked about with gene rearrangement early on to create the B cell receptors and the T cell receptors. Except for now, this is happening within the constant regions. So the areas that need to be switched are identified, the intervening DNA is looped out and excised and eventually degraded. The idiotype is then joined to the new constant region, and that's going to give us a new isotype of antibody. This is sort of a one-way event. So it's not like, ladies, when we go shopping, we find that pair of shoes that we love and then we take it home and take it out of its lovely shoe box. We put them on and we realize we can't walk 2 inches in them. So we take them back, right? We can't do that here. It's like there's no buyer's remorse here. So once we switch, we're sort of stuck with what we switch to.